Hi everyone, my name's Donna Gairns and I'm the National Nurse Director with Lymphoma Australia, and I'm joined today with Associate Professor Chan Cheah for Sir Charles Gairdner and Hollywood Private Hospital in Perth, and he's also the Director of the Blood Cancer Research in WA as well. Welcome, Chan.
Hi Donna, thanks for having me.
Thank you for joining us. So this week we're doing a series of interviews with Australian lymphoma experts across the country, as we'll all, Lymphoma Australia and a lot of us were supposed to be in Frankfurt in Germany this week for the annual European Hematology Association, or EHA Congress. But due to COVID-19, as many other things, and associated travel restrictions, we're all in Australia and been watching this fervently on the online, and it's great to be able to still give you the updates of what was presented through the conference. So thanks, Chan, for bringing some of your time and your expertise to discuss some of the key abstracts that you found quite interesting through the conference and you're watching. So we thought we'd just start off with the first one. We thought we'd talk about the Aspen Trial a little bit. That was done by one of the lead doctors in Australia, is Professor Chan Cheah from Peter Mac. And this study is a phase 3 study for patients with Waldenstrom's macroglobulinemia. It's a big word, that one. Yep. So just want to give some overview.
I remember to turn my phone to mute beforehand, Donna.
Yeah, I should have. Sorry about that, everyone.
That's okay. So yeah, look, the Aspen trial was, we've all been waiting for the results of this for some time. And this trial opened about 3 years ago from memory. Australia played a big part in enrolling. Australian patients were really important in getting this study done. And it was open at around about 6 or 7 sites across the country. As you mentioned, Chan Cheah at Peter Mac was senior author on this trial, and we also enrolled quite a large number of patients at our site in Western Australia. So this was a phase 3 open label study in which 2 BTK inhibitors, brutonib tyrosine kinase inhibitors, were compared head to head. One drug was the standard of care, ibrutinib, and the comparison was a new drug called zanubrutinib. This drug was previously called BGB-3111, and much of this drug's development has actually taken place in Australia. Zanubrutinib is one of what's called the second generation of BTK inhibitors, they're called second generation because the number of enzymes that it blocks appears to be lower than ibrutinib, so they're felt to be cleaner drugs with less off-target side effects. And the point of the trial was to see whether people who had Waldenstrom macroglobulinemia, which is a common type of low-grade, actually it's uncommon type. It's common at our site because we have so many trials running, but overall it's a relatively infrequent type of low-grade lymphoma. The unique thing about Waldenstrom macroglobulinemia is that patients often have a detectable protein in their blood called IgM, which you can use to monitor response. Now, in this trial, patients received either the standard drug, ibrutinib, or the new drug, zanubrutinib. There was a 1-to-1 randomization, which meant it was a 50-50 chance of getting either drug. And the point of the trial was to see which was more active or more effective, and also what the safety profiles were like. It was designed to show superiority for the new drug, and the bottom line is that it did not, in the sense that the effectiveness of the 2 drugs appeared to be similar. And that meant that the control of the Waldenstroms, in terms of the number of people who had a favorable response and who had a reduction in their proteins and a shrinking in their lymph nodes, was similar between the 2 arms. However, there were differences in the safety of the 2 medicines. You know that ibrutinib is associated with a few side effects which are specific to BTK inhibitors or BTK inhibition, like bruising, and certainly that was seen in zanubrutinib as well. But atrial fibrillation, which is a fast irregular heart rate, which can cause palpitations and shortness of breath, we have noticed that seems to be an issue in some patients who take ibrutinib for long periods of time. And the rate of people getting atrial fibrillation on ibrutinib can be up to 1 in 6. And in the new drug, zanubrutinib, there was much less atrial fibrillation seen. I think it was about 2 or 3%, lower than about 15% on the patients who got ibrutinib. So there was a fairly significant difference in safety profile there. So when you consider that there's lower rates of some of those less desirable side effects, despite the fact that overall the trial showed equivalent activity, because of the more favorable safety profile, it's probably going to make a lot of people prefer zanubrutinib over ibrutinib, given that they appear to be just as effective each other. Now, whether that translates into the regulators giving an approval for funding for zanubrutinib is an entirely different issue, and we have to wait for the outcome of that. So that was that trial.
Okay, that sounds really promising, especially the less toxic effects of the drugs and stuff.
I think it's really important for patients. It's one thing being able to control the lymphoma, but it's another thing having to live with these medicines for a long period of time. You take these until they stop working. So I think side effect profile is really important.
Definitely. And zanubrutinib at the moment, it's only in clinical trial in Australia, isn't it?
That's correct. It's approved in the USA for relapsed mantle cell lymphoma. In Australia, because a lot of the development's taken place here, it's available on clinical trials in most large centres, but not available. It's not reimbursed yet.
Okay, great. Thank you for that. We'll watch and wait and see what happens with that. The second study we thought we'd talk about is for another rare group of lymphomas, mantle cell lymphomas with CNS disease. There was a study that you're a co-author on. Would you like to give us a rundown on that one and what was presented?
Sure. So look, it was really good to be part of this. In fact, my involvement with this particular niche area, this dates back to 2013 when I was working with you at Peter Mac, actually, Donna.
Okay.
And, we looked at a large number of patients, about 50 or 60 patients who had mantle cell lymphoma where it had spread to the brain or spinal fluid, which is a rare but very difficult to treat event, as it is in all lymphomas. At that time, the standard, all we had was chemotherapy. And unfortunately, when any kind of lymphoma, and mantle cell lymphoma being no different, when it gets into the brain, it's very difficult to treat. And we found that the outcomes from treatment were not great. And so that was 2013. More recently, we collaborated with an Italian group led by Dr. Chiara Rusconi, and she had collected a group of patients with mantle cell lymphoma who had central nervous system involvement, who were treated with ibrutinib, which we discussed earlier, a tablet, BTK inhibitor. And in her preliminary data set, it appeared that ibrutinib seemed to be pretty active. And we know that ibrutinib has some ability to enter the brain, unlike some other medicines. The level of ibrutinib that gets into the brain and the spinal fluid is about 7% of what it is in the blood, which is often enough to have some benefit. And we know that from small case series, ibrutinib has been reported to have some activity in mantle cell lymphoma in the brain. So in this study, we got together about 80 patients with a central nervous system involvement for mantle cell lymphoma, which is quite a large number for a rare event in a rare type of lymphoma. And we looked at people who'd had ibrutinib and people who'd had any other kind of treatment. And the conclusion was that patients who got ibrutinib appeared to have a better likelihood of the lymphoma going away, and they lived for longer. And that was compared to all other treatments lumped together. So all kinds of chemotherapy, radiation, steroids, all the rest of it. That the main difference was if you had ibrutinib, then you had a better outcome with. So that's really, I think, an important finding, because we'll never be able to do a randomized clinical trial, which is the highest level of evidence, in this group of patients, because they're so rare. You could never do a study. So this kind of looking backwards study is pretty much the only way that we can answer this question. And so as a consequence, I think that people are going to look at this data set and say, well, clearly, if I have a patient with mantle cell lymphoma and they get it goes into the brain, then ibrutinib or some sort of BTK inhibitor
Yeah.
should be preferred over giving high-dose chemotherapy, for instance. So I think that's practice changing, even though it's not a clinical trial. I
Yeah.
think that it's a practice changing abstract.
Yeah, and you're right, and it's because it's so rare. It's very hard to capture these patients as well, and it's
Yeah.
great you've had so many patients you have been able to capture over that time. Do you think this would be able to translate into other CNS lymphomas as well with using ibrutinib? Or
It's
have
a good question. I mean, we know that ibrutinib has activity in primary central nervous system lymphoma, and there have been a number of trials that looked at that. In fact, my fellow Catherine Lewis just published an observational data set looking at lymphoma in relapsed primary and secondary central nervous system lymphoma, which is another difficult sort of situation to treat.
Yeah.
And again, there does appear to be promising activity there. So I think that the ibrutinib certainly needs to be looked at further in lymphoma, which is spread to the brain. And the one difficulty that we're going to see increasingly is many of our patients with mantle cell lymphoma are now treated with BTK inhibitors as part of first or second line therapy on trials or as standard of care. And it's not clear. And what this study does not tell us is what we should do with people who develop central nervous system involvement when they've already had a BTK inhibitor. Because that's an unanswered question. It's happened to me once before. Actually, it's pretty hard to treat.
Yeah, no, it sounds pretty difficult, that's for sure. And just the third study we're going to be talking about today is just the updated results with the OASIS trial that was presented by Professor Stephen Legould with newly diagnosed mantle cell lymphoma. And I had the opportunity of speaking with Stephen about this trial at ASH, but could you please give us a bit of an update on that presentation?
Of course. So the OASIS study is a phase 1b combination of obinutuzumab, venetoclax, and ibrutinib in patients with mantle cell lymphoma. So a non-chemotherapy triplet combination, all active drugs against B-cell lymphoma, venetoclax and ibrutinib particularly so. You recall that Stephen has previously presented the earlier arms of this study in which patients with previously treated mantle cell lymphoma received the triplet. In this presentation, he was giving an update on 15 patients who had never had treatment before. And he looked at and as you'd expect, patients tended to do very well. The response rates were very high. The side effect profile was pretty acceptable. There wasn't anything unexpected that you wouldn't have thought you would see from these combination of drugs. And so it seemed to be certainly fairly easy to deliver. The interesting thing was when he actually did a comparison of how deep the responses were compared with previous arms of the studies. And what we saw was that for patients who'd never seen chemotherapy before, the drugs appeared to work better. And that probably is not surprising because the more chemotherapy you've had, the more resistant your lymphoma cells are to treatment. And if you take a group of patients who've never been exposed to chemotherapy before, they're likely to respond better to any kind of treatment, but in particular, these sort of biological or non-chemotherapy strategies. So when he looked at the depth of response, and that was measured by using a molecular technique called minimal residual disease, he was able to show that patients who had never seen chemotherapy before had a deeper response, and there was much lower levels of lymphoma detectable than in people who've had been treated previously. And that raises the question, well, maybe we should be using these kinds of combinations very early on in treatment instead of at the fourth or fifth line of treatment because we tend to get better responses. So that was very interesting.
Yeah, that sounds really good. Do you remember how long the follow-up has been so far for these patients?
Pretty short. From memory, I think that the follow-up was somewhere in the vicinity of 12 months or even under, So we don't really have durability. We don't have information about the durability of these responses. And clearly that's going to be a key question. And when you consider that the existing standard of care for patients with younger patients with mantle cell lymphoma at the moment, things like the Nordic regimen and CHOP, alternating, RDAP and transplant and things like that. We're seeing, on average, people going for 7 or 8 years before they have their lymphoma come back. So the key thing to remember is any changes that you try and make to frontline treatment will really need to have a good look at how long they last for before we start and doing randomized trials comparing them to existing standards of care. Because although chemotherapy has a lot of side effects, we know that in most people, it actually controls for lymphoma for quite a long time. There will be people with high risk, a high risk subset of patients with mantle cell lymphoma, where using non-chemotherapy approaches will be highly desirable. And I'm thinking here of patients with TP53 mutations or TP53 deletions, which are about 1 in 10, 1 in 20 patients with mantle cell lymphoma. Yeah. And in my practice, if I find these things up front, then I'm pretty much universally streaming them to some sort of clinical trial novel agent option rather than using chemotherapy. And I think that Stephen's data, in fact, the interesting thing about this study was that he actually had quite a few high-risk patients in those 15, and that they seem to do pretty well with responses as, very small numbers, but it seemed like even though there were some high-risk patients, they did well. So that's very promising. And, Contam showed the same thing in the AIM trial, which was that combination of venetoclax and ibrutinib in mantle cell lymphoma. And he showed that I think 5 of 12 patients on that study who had problems with TP53, which is a high-risk genetic mutation, actually ended up having complete responses, which is something that we certainly don't see with chemotherapy.
Mm-hmm. Is it standard practice for all doctors in Australia to let patients know when they're diagnosed with mantle cell lymphoma to get these molecular testings done?
No, it's not.
Yeah.
Ideally, it should be. The problem is access. Yep. The testing is not reimbursed at the moment, and so we're fortunate at my site that the state government will just pick up the tab for that testing and absorb it. But unfortunately, the equity and the access to molecular testing for things like TP53 and mantle cell lymphoma is very much driven by the interest of the clinician and the pathologists in working that out. So we're very fortunate that we have a group of pathologists who are very interested in lymphoma at my site and a group of clinicians who are very interested in lymphoma. And so we routinely will do it. And I would like to say that it's done everywhere. And I think increasingly it will become a more widely done test. But the unfortunate fact is, Donna, that if you don't have access to a clinical trial, then it's not necessarily going to change your frontline treatment. Because if you take clinical trials aside, then if you look at what's on the PBS at the moment for frontline for mantle cell lymphoma, then whether or not you have TP53 doesn't actually change what you do if you don't have access to a clinical trial.
Yeah. This is definitely something for people to think about when you are diagnosed and to ask these questions too.
I think so. I mean, if I was diagnosed, or if my mother was diagnosed with mantle cell, father, sorry, it's more of a male disease, was diagnosed with mantle cell lymphoma, then you definitely want to know what your TP53 status is. I mean, there's a few ways that you can look at it, but next generation sequencing and FISH are two molecular techniques which can be used, and many labs have the capability of doing this, but it does cost a bit of extra money. Yeah. I think if you push your hematologist to get it done, usually even if they can't get it done at that lab, you can often send it to another lab and it can be done elsewhere. So for instance, Peter Mac, I'm sure that the pathology team at Peter Mac do TP53 mutation and deletion testing as a matter of course, as do we. And so you find that your big laboratories at quaternary academic centres will tend to have that capability. And but as I said, I mean, the key thing is at the moment pushing clinical trials as the way to go. If you've got a TP53 mutation, I would certainly be trying to access a clinical trial. For instance, NHL 33, Eliza Hawke's study with acalabrutinib rituximab for transplant eligible patients with mantle cell lymphoma. If I had a TP53 mutation, I would go on that over having standard of care, because then you're getting that novel agent in frontline. And similarly, if you're transplant ineligible, then there are a number of trials around the country. We've got several enrolling BTK inhibitors in frontline. So if I find P53 mutations in my patients who are over 70 and who have mantle cell lymphoma, I don't give them BR. I put them onto trials evaluating new BTK inhibitors.
Yeah. That's a great note to note, and people can always contact Lymphoma Australia and we can help
Of course, yeah.
option too. So
Absolutely.
thank you so much, Chan. That was a great update, and I'm hoping next year we might be able to be at the actual conference.
Yeah, it's well, next year is going to be a Lugano year, so we'll have to see. I'd hate to miss that.
I did not miss that. Okay. Thank you so much. Thank you, everyone.
Pleasure,